Hi there, my name's Rose Hodgson and I'm a research scientist in the immuno-oncology team here at MiNA Therapeutics. Here I'd like to present to you today compelling data from the application of a small activating RNA oligotherapeutic, MTL-CEBPA, which has demonstrated favourable clinical response in cancer patients with hepatocellular carcinoma, leading to enhanced tumour regression. It's well known that the tumour microenvironment of the solid tumours promotes the recruitment and polarisation of key immunosuppressive myeloid populations, such as tumour-associated macrophages polarised to the M2 phenotype, or also immature myeloid-derived suppressor cells that circulate in cancer patients' peripheral blood. These immunosuppressive myeloid populations in turn can promote a tolerogenic phenotype, which can lead to immunological ignorance of cancer cells, tumour progression, and it can lead to treatment resistance. A key current strategy of the immuno-oncology world is targeting these immunosuppressive myeloid populations in order to promote a favourable TME and therefore improve therapeutic efficacy. One key transcription factor called CEBPA is known to be important for myeloid differentiation, and key papers have demonstrated that it's reduced in several solid tumours. So here enters MTL-CEBPA, which is a small activating RNA targeting the CEBPA gene that is formulated within the NOV340 liposomes, which are myelotrophic, and this is formulated together to form the drug product known as MTL-CEBPA. Understanding the cell types where MTL-CEBPA is delivered is key. Here we show the functional biodistribution of the liposome within bone marrow CD34 populations and also monocytes in the peripheral blood. Here we show that this is the myelotrophic drug, and this is an important finding for the mechanism of this small activating RNA therapeutic. Currently, MTL-CEBPA is in clinical trials in cancer patients with hepatocellular carcinoma, and this slide highlights key results from a phase 1 clinical trial of MTL-CEBPA in HCC patients also treated with standard of care, highlighting significant anti-tumour activity and tumour regression as shown in a complete responder patient here. Furthermore, the clinical response of these HCC patients treated with MTL-CEBPA correlated with post-treatment upregulation of the key target gene CEBPA, indicating potential use of this upregulation of CEBPA as a biomarker of clinical response. And almost unsurprisingly, upregulation of this key immunological and immunomodulatory transcription factor listed vast key gene expression changes in the peripheral white blood cells of these HCC CEBPA treated patients, highlighting several potential biomarker genes, such as I'll highlight here IDA1, which is reduced in day 2 after treatment, and this gene is known to be associated with immunosuppression and MDSC presence. Furthermore, treatment with MTL-CEBPA also elicited changes in the peripheral cell populations known as PMN, which is polymorphonuclear, and monocytic MDSC populations in the peripheral blood of these HCC patients. And it also led to a reduction in the intratumoral M2 polarised CD613 positive macrophages post-treatment of these in a complete responder patient, as shown here by the immunohistochemistry plots here. Together, this leads us to propose the mechanism of action for MTL-CEBPA, where it leads to reduced MDSC numbers in peripheral blood and also reduced immunosuppression via M2 TAMs intratumorally. These changes also suggest that MTL-CEBPA treatment reduces global immunosuppression, leading to increased therapeutic efficacy of conventional anti-cancer drugs and clinical responses described here in HCC patients treated with MTL-CEBPA and standard of care sorafenib. Here we can take away that MTL-CEBPA is a safe, well-tolerated, first-in-class oligotherapeutic with clinical efficacy in HCC. We now progress to a phase 2 study of MTL-CEBPA in hepatocellular carcinoma. Single-cell RNA-seq of immune populations across hepatocellular carcinoma identified that CEBPA was lowly expressed in macrophages in the HCC TME. Furthermore, when we performed a similar analysis across solid tumour types, TME immune populations, we could see that CEBPA is also lowly expressed in M2 populations there, as you can see by this graph here. This indicates potential treatment avenues for MTL-CEBPA across tumour types as a well-tolerated therapeutic that can reprogram the immunosuppressive TME and create a permissive space for standard of care combinations. Thank you for your attention. We look forward to any questions, and please note that MiNA Therapeutics is open to collaborations of any kind. Thank you.